Alright, these are your notes over translation. The essential question, how are amino acids assembled in the second part of protein synthesis? Translation. This is our outline, protein synthesis. We're in part 2, translation. This is what we've been doing on the board. And then that was our video that we've been watching in YouTube. Protein synthesis overview. Be able to label all of this good stuff with these words and draw those guys in. What's this part called? Yep, transcription, and then the second part is, okay. Part 2 is translation. It occurs in the ER. It also can happen in the cytoplasm. If you have free ribosomes in the cytoplasm, you take the message that we just made, the mRNA, and make the protein. It is the second part of protein synthesis. Okay, here we've got a Russian guy and a Chinese lady, and we are translating, the language. And specifically in the ER, this is going to happen in the ribosome. Okay, translation, step by step. Step 1, we've got the mRNA, and it carries the message into the ER. So bam, this little guy that we made is our message. Number 2, it enters the ribosome, the blue guy right here, the protein maker. Number 3, the ribosome reads the message, the mRNA, and the specific tRNA comes in. So my green guy is my tRNA. The tRNA has a specific amino acid on top. Bam. We've got tRNA with the amino acid. And then steps 3 and 4 repeat. So another tRNA with another amino acid. Another tRNA with another amino acid. The amino acids form a chain, which is the protein. So this guy right here is going to be the protein. If you notice, there is the codon on the message is color-coded for the amino acid that it brings in. So we have dark blue, light blue, blue, and then green, and then way back at the beginning it was red. You guys got all that? The circle chart is how we read the genetic code. The purpose is to determine which amino acid is needed. It's very important. You must use the 3 letters of the message, of the mRNA. So the 3 little letters on this guy, not on the tRNA, because that is our message. That's like our code. All proteins are going to start with met and end with stop. I'm not sure if you've been noticing that from our worksheets. So you're going to go start in the center. So if it was AUG, you would go A, and then out to U, and then G. So we've got met. See if you can do those 3 without looking, and see which amino acids you've got. Alright, this is the last part, codon and anticodon. The codon, it's the 3 letters on the message, the code, right there, the codon. And the anticodon, anti means opposite, the 3 letters on the tRNA. So my little T has 3 little letters on it. And those are going to bond and bind by our rule. Remember, the nitrogen bases are, for DNA, we have ATGC at Garden City, and then the RNA as AUGC. And for the RNA, we are talking about mRNA and tRNA. So both of those guys are going to have that U in it. Alright, do your Quizlets and summary, and we are good.